Hello students, welcome to Aakash. I'm your botany faculty Dr. Sukirti, and today what we are going to discuss is mock test number 7 of AIATS Plus 2025 phase 2, which was held on 2nd of March 2025. So in this particular session, we are going to deal with certain chapters, right, which are important with respect to the NEET examination, and we are not just going to solve the questions. Apart from that, we are also going to know about the entire periphery of that particular topic, right? So the chapters that we are going to discuss in this particular session would be principles of inheritance and variation, cell cycle, microbes, ecosystem and respiration, right? So let's quickly move ahead to the very first question. The very first question over here, it says that in genetics experiment, Punnett square is, right? So this particular method was given by Quinel C. Punnett, right? Now, in order to know about it more, firstly what we should do is to know about how to perform a cross. So we know for a fact that when the genetics, the foundation of the genetics has been laid upon by Mendel, he performed certain crosses, right? Like monohybrid crosses, dihybrid crosses and so on. So we are going to start with the monohybrid cross first, where we are going to take a single character into consideration, and let's take the standard one, that's the plant height, and we are going to talk about a typical Mendelian cross, right, in which we are going to talk about the plant height in the Pisum sativum plant. So if we say initially when we take parents, so let's say the pure line parent having tall stem height is crossed with the pure line again having dwarf stem, right? Now if we make gametes from them, a single type of gamete will be produced by them, right? So how we propose that, how many types of gametes would be formed? We could calculate it with the formula 2 to the power n, where this n represents the heterozygotic pairs. As we could clearly see over here that in this case or in this case, in both the cases we have homozygous parents. So this n value comes out to be 0, and that's why the answer will be 1. So there will be single type of gamete that will be produced by these parents. Now when they fuse together, a heterozygotic progeny will be produced, right, in F1 generation. Further, in order to have the F2 generation, we'll perform a self cross. That means crossing the individual with another one having the similar genotype, right? So this is the selfing we are doing. Then further, of course, gametes will be made by these also. So it will be capital T, small t this time, because there is one heterozygotic pair now. So 2 to the power 1 makes it 2, right? So same goes for this as well. Now if we make the Punnett square like this, right? From both the parents, we'll write the gametes like this. So we'll be having these kind of genotypes. This will be tall parent, this will be again tall, this will also be tall, and this is going to be dwarf, right? So this is the F2 generation. So what we could infer from here about the Punnett square, that it is talking about that what kind of genotypes are possible if we perform a cross. So these are the possible types of genotypes that we are going to observe in the progeny. It is not anyway talking about the total number of progenies that we are going to get. We are going to decide the type of genotypes, right? Now with this information, let's get back to the question. And over here, the very first option is talking about, it is used to calculate the number of offsprings. So that cannot be, analyzed with the help of Punnett square. If you want to calculate the number of offspring, you have to go for the absolute count when you are performing a cross, right? You literally have to count each and every number of organism or progenies that are being produced. Then second says it's a graphical representation to calculate the probability of all possible genotypes in a genetic cross. That's what Punnett square is used about. We can identify what type of genotypes we are going to observe in the progeny. Also, we can calculate the probability. Like, if you want to calculate the probability of heterozygous individuals in the F2 generation, that would be 2, because 2 individuals are there out of 4. So that makes it 1 by 2, 50% or probability of having these progenies in the progeny of F2 generation, right? So this is how we could use the Punnett square for. Now third one is saying it is required to know the cause of dominance of alleles. If we really want to know that why a particular allele is dominant, we have to perform the genetic analysis or a biochemical characterization of it. That what are the possible clauses or explanations that this particular allele is dominant over the other one, right? Not the Punnett square. And the fourth one is saying that essential to know the reason of incomplete dominance. This is again incorrect. In both the cases, we are already presumed the fact while making the Punnett square that whether the gene is exhibiting complete dominance or is it exhibiting the incomplete dominance. It is nowhere explaining the cause of it, right? So that is how we can clearly infer that in a genetic experiment, Punnett square is basically used to calculate the probability of all possible genotypes in a genetic cross, right? So this is how you will be solving this particular question. I hope this is clear to everyone. A very good morning to everyone. All right. Now let's get back to the next question, and that talks about law of dominance and law of segregation was given on the basis of the observations of. So when we talk about the Mendelian genetics, he basically proposed 3 laws: the law of dominance, the law of segregation, and the law of independent assortment, right? So if we talk about the law of dominance and the law of segregation, how we could infer it. So when he proposed this law of dominance, he proposed certain facts, like when there will be a character is going to be controlled by a single gene, every gene will be having alleles in a pair, and then if an allele is capable to express itself in all conditions, whether it's a homozygous condition or a heterozygous condition, then it will be termed as the dominant allele. And if on the contrary, if it is only going to express itself when there is an absence of dominant allele, that means in the homozygous condition, then we are going to call it as recessive allele, right? At that time, he didn't use the word allele, he used the word factor. But in order to understand that, we are using the term allele here. So if we get back to this cross, the monohybrid cross, if we have to ascertain the points that are being proposed by Mendel, that allele will occur in pairs, right? We could clearly see over here that they occur in pairs. Then secondly, the allele that expresses itself regardless of what other allele is going to be, so that allele is going to be termed as dominant allele. In this particular case, we could see that this one is going to exhibit tall phenotype. Same goes here. Here also, if capital T is present, it's irrelevant that what's being present in the second position, the phenotype is going to remain tall only, right? So this is how we can express that the T allele is dominant, because irrelevant of what is the other allele present in the pair, it expresses itself and give the tall phenotype. On the contrary, if we see the small t allele over here, it is expressing itself only when there is an absence of the dominant allele, or we could say in the homozygous condition. If that is present, only then it is expressing itself, and that's why we are going to call this one as the recessive allele, right? So from the monohybrid cross only, we could identify law of dominance, right? The next law that says law of segregation. So law of segregation says that during the time of gamete formation, the allelic pair is going to segregate, right? So one gamete is going to receive the one, part of the allele. Like for example, over here in a pair, we have two alleles, capital T and capital T. half of the gametes is going to have this and half of them is going to get this allele. But over here we are not going to analyze it really clearly because all of the gametes that are going to be formed is carrying capital T allele, right? So in order to differentiate that and understand that how exactly the equal segregation of allele is occurring, we need a heterozygous genotype, capital T, small t. So with this, half of the gametes are going to carry the capital T allele while the other half is going to carry small t allele. That's why they have two types of gametes over here, one with capital T and another with small t. So this shows that segregation occurs during gamete formation, and that was the proposition of law of segregation. So with this cross only, we have identified to propose both law of dominance and law of segregation. So if we get back to the question, it says that on the basis of the observations of which type of cross, law of dominance and law of segregation was proposed. So it was because of the monohybrid crosses. If we talk about the dihybrid crosses, it helped in the proposition of law of independent assortment. Which talks about the fact that one gene pair or allelic pair is not going to affect the transmission of the other one, right? That is why it is possible that, like when he performed the cross with round seed and round and yellow seeds with wrinkled and green seeds, he identified that even the round seeds can become wrinkled, right? Even the round seeds can become green, and even the wrinkled seeds can become yellow. So there is every possible combination that could be there is present when the F2 generation was being observed. So that is how he proposed that one gene pair never affects the transmission of the other one, right? That means the shape is not affecting the transmission of the color in the seeds of Pisum sativum. So then trihybrid cross, this particular cross is used to ensure the fact that law of independent assortment still holds true, or the law of dominance still holds true, law of segregation does holds true. So these trihybrid, tetrahybrid crosses were being performed in order to statistically satisfy these three particular laws. Now talking about, so this is again incorrect, this is also incorrect, and fourth one is back cross. So what is a back cross? Back cross means when a progeny is being crossed back with the parents. Like for example, if let's suppose we are having the classical cross with the plant height as we talk about, capital T, tall plant is being crossed with the dwarf one having small t genotype. Then of course they'll make gametes. This will be capital T, this will be small t. After the fusion, they are going to produce this particular progeny. Now, if I cross this particular progeny with either of the parents, that means if I am performing this cross or I am performing this cross, both of them are being considered as back cross, because over here the progeny is being crossed back with the parent, right? So in this case, what we are going to have, capital T, small t, here's capital T, small t, right? This type of cross is known as outcross, where, so outcross, and this you can clearly see that this is an example of a test cross. So we can say that test cross is a type of back cross, right? Where the individual is being, this particular cross is also exemplifying the test cross. So over here, the progenies that we are going to obtain would be capital T, small t and small t, right? So this is how a back cross is there. So back cross, he didn't perform the back cross in order to propose these, right? Because back cross could be monohybrid, dihybrid, tetrahybrid. So the minimal amount of characters that are helped in the proposition of these two was the single character, put forward in the monohybrid cross, right? Is it clear to everyone? All right. So yes, Madhushri, you have answered it correctly. The option 1 is the correct answer for this question, right? All right, now let's move ahead to another question. This says that we have to select the correct set of parents, those can produce child with blood group O. So when we talk about the human ABO blood group system, so we know that human ABO blood group system shows multiple effects, such as it shows complete dominance, right? It shows codominance as well. And another thing it shows is multiple alleles. Now, what are multiple alleles? When we'll be having more than 2 alleles for a single character. Unlike the Mendelian laws, which says that for every character there will be an allelic pair, right? So when we talk about human ABO blood group system, it has 3 alleles, IA, IB and small I. If we talk about the order of dominance, these two are equally dominant and they both are dominant over small I, right? Now we know for a fact that we can have 4 types of blood group in humans, that is A, B, AB and O. If we talk about the genotypes, we can have for A blood group these genotypes. For these we can have IB, IB and IBI. Here we can have IA, IB. Here we can have small I, right? Is back crosses possible in human genetics? No, that's not. We could only perform it with the plants, not with the human, right? Ethically also, and obviously it's not feasible to perform the back cross in human genetics, right? All right, so going by this, if we see these genotypes IA, IA and IAI, as we have discussed the previous crosses, we could clearly see that in this case IA gene is expressing itself whether the other IA allele is present or small I is present. So this is a case of complete dominance, right? That means a dominant allele is capable to express itself both in homozygous and heterozygous condition. Same holds true for this case also. When IB allele is present with another IB or small I, again the B blood group is going to be there. So this is also a case of complete dominance. Now coming to this IA and IB. So over here the blood group is AB. That means both the alleles are expressing themselves equally. So this is a case of codominance. And it is of course the exception of the Mendelian law of dominance, which proposed the fact that dominant allele will always express itself, right? And whether it is regardless of the fact that what other allele is present, only one dominant allele is present for a character. Over here what we are seeing, both of them are equally dominant, and that's why this is a case of codominance. And this again small I, case of dominance only, right? Kyunki over here what we are seeing that when recessive allele is present in the homozygous condition, it is expressing itself and giving the blood group O, right? So this is how human ABA blood group system exhibits lot many inheritance patterns. Now getting back to the question, what we have to do, we have to select the correct set of parents which can produce the child with blood group O, right? In this particular case, what we have to do is to identify that whether both the parents are going to carry the small I allele or not, right? Because the progeny that is going to be formed will be formed as a result of fertilization. That means gamete from both the parents are going to get fused, and that's how the zygote will be formed and later on giving rise to the child, right? So if both the parents are possessing small I allele, only then the progeny is going to get the O blood group, right? So if we see over here in this parent IA, I and IA, I, right? They'll make two types of gamete. So if these two are going to get fertilized, we'll be having the production of O blood group. So yes, definitely this is going to be producing an O blood group child. Now going with this, if we have IBI and IAIA, so they are going to produce two type of gamete, IB small I, IA. In any of the case, any fertilization, if this fertilize with this, it will be producing AB blood group. If this fertilizing with this, it will be having A blood group, right? So that is why this is not going to be formed. Now in this particular case, in case of C, again both the parents are carrying the small I allele. So of course O blood group is going to be produced. In this case, this parent is lacking this allele, that's why it's not going to be there. So A and C are the ones which are going to produce the progenies having the O blood group. So yes, option 1 is correct. Yes, Madhushri, wonderful that you have identified it earlier also, right? But we are trying to identify the concepts behind it, right? So wonderful. Now the first one is correct and we could rule out all others over here, right? So this is how you will be performing the cross, or basically just identify the type of gametes they are going to produce, right? Okay. Now let's move ahead to another question which says that we have to read the following statement and we have to choose the correct option, right? Over here statement A says that dominance is an autonomous feature of a gene or the product that it has information for. So this statement is incorrect. Why? Because when we talk about dominance, it's not only going to depend upon the gene, it's also dependent upon the product, the condition in which it has been produced, and various other sequences that are being present prior to it, or the body conditions, right? So it is not the autonomous feature of the gene, right? On the contrary, over here in this statement it has been written that it is an autonomous feature of a gene. Also, when we talk about dominance, we may find that in one case a particular gene is being dominant for a particular character, but for some other character it's being recessive, right? Or not having the ample amount of dominance. Maybe it shows incomplete dominance. Like for example, if we consider a gene like this, B gene, if we consider, let's say, and it's controlling the starch grain synthesis in case of Pisum sativum. So if we talk about the starch grain size, right? If this particular genotype is there, it's going to produce large starch grain, right? If it is having this particular genotype, the heterozygous form, so then it will be intermediate starch grain. And if it has small b, then it will be small starch grain, right? But this gene also controls another effect, that is the seed shape. So in these two cases, it is going to show round seed shape, and in this case, it is going to show wrinkled one. So what we are seeing over here, that for one character, that means the starch grain size, this particular gene is showing incomplete dominance, and in case of the seed shape, it is showing complete dominance. So it's not dependent upon the gene, what is being written over here. That's why we are inferring the fact that it is an incorrect statement. Now statement B says some character in pea plant can show incomplete dominance. We have seen here just the example that the starch grain size, if is that being considered as a character in a pea plant, it shows incomplete dominance, as when the heterozygous genotype is being present. In that particular case, the size that we are obtaining is an intermediate sized one, right? As opposed to the law of dominance, which proposed the fact that if the dominant allele is present, then definitely there will be a complete expression of it, right? Autonomously express ही होता है पर कुछ problem होने पर नहीं हो पाता. See, it's not about the autonomous expression. I am saying that autonomy means that regardless of any condition, it is going to express itself in the same form, right? So that is the reason why, if I take this particular example, single gene is carrying out two different effects, and in both of them it has different type of expression. So that is why we cannot propose it's an autonomous feature of a gene, right? For different consequences, for different characters, it is going to be different. Sometimes it's also dependent upon the need of the body. Like for example, at a particular moment of time, energy is being needed, so those particular genes which are expressing for the enzymes which are going to carry out the mechanism of aerobic respiration is going to be, enunciate more. In some other time when it's not being needed, it's being suppressed. So we are not going to say that dominance is going to be autonomous feature of a gene, because if the body does not need the expression of those genes, they are going to remain silent, not going to express themselves, right? Okay, so is it necessary to read out of NCERT for biology? Like, it's not that case. NCERT driven till now, what we have observed in the PYQs, they are already NCERT based. It's just that we have to thoroughly understand it, right? That's what the key is, that you have to thoroughly understand the NCERT, right? All right, wonderful. So as we could see here, so this is a correct statement, and that's why we could simply say that only statement B is correct, correctly depicted in option 4. So Ankul, Khushi, wonderful, you have already identified it. So option 4 will be the correct answer for this particular question. Okay, moving ahead, we have another question which says question number 127, which talks about a pink snapdragon flower is crossed to a white snapdragon flower. What's the probability of getting a red snapdragon, right? So when we talk about the snapdragon flower color, it basically exhibits incomplete dominance. Right? So in incomplete dominance, what happens is that the dominant allele is not capable to express itself completely in case of heterozygous condition, right? So if we talk about the snapdragon flower color and we start with the cross, we'll be having, let's say, pure line. This is the red color crossed with white colored flower. The gametes they are going to make would be capital R and small r, and when they fertilize, they are going to get capital R, small r. These progenies, unlike the law of dominance, is going to exhibit not the dominant phenotype, rather an intermediate one. That's the pink color, right? This is the F1 generation. And this is how we could define incomplete dominance, that in heterozygous condition, the intermediate phenotype is coming in. So getting back to the question, it says that there is a pink snapdragon flower, right? So that means the parents are already being defined. We have pink snapdragon flower, so that means it's going to be heterozygous genotype, right? And it's being crossed with the white snapdragon, that is small r, small r. So the gametes they are going to produce would be capital R, small r, and again small r over here. So if these two fertilize, we are going to have capital R, small r. This we are going to fertilize, then it will be small r, small r. So we have obtained pink and white in equal proportion, right? Pink and white in equal proportion. So we are not having any red snapdragon. So of course, the probability of having a red snapdragon would be 0, correctly depicted in option 4. So Ankul, Amit, Ashish, wonderful. The option is going to be 4. One more thing. If I ask you that whether this particular cross that's been written over here is an example of test cross or not, just put on the comment box that whether you feel that this particular one is a test cross or not. Wonderful, Ashish, that's already been solved. Kudos to you. Yes, it's option number 4. But what I'm asking is that could you consider this as a test cross or not? See, when we say about the test cross, we always say the fact that test cross is used to identify the genotype of the test organism, right? So the genotype is being identified after performing the test cross, right? So if you're saying that it's an example of test cross, that means we don't know the phenotype of this particular, the genotype of this particular plant having pink, color. See, in snapdragon case, flower color case, as it is showing incomplete dominance, we know for a fact that if a flower color is coming out to be pink, the genotype that it is going to have would be heterozygous, whether we perform this cross or not, right? So that is why we cannot exemplify this as a test cross, because we already know the genotype without even performing the cross, right? So that is why this is not being considered as a test cross. All right, I hope this is clear to everyone. Okay, so let's move ahead with another, question. And now this question says about that detritivores break down detritus into smaller particles by a process known as. So when we talk about that detritus, break down detritus into smaller particles, so it is the process of decomposition that we talk about, right, in which the very first step, that means when we talk about the detritivores, they act upon the detritus, right, and convert it into a smaller fragment, and that particular process is known as fragmentation, right? The bigger substances broken down into the smaller pieces. Correctly said by Ankul and Amit. Wonderful. So fragmentation is the process. Now why this process needs to be done? Because when we talk about the microorganisms which are even smaller, they cannot able to access this much big detritus initially. So in order to simplify the process and in order to obtain nutrition as well, detritivores further simplified this process and make those bulky chunks of organic matters into simpler substances. First, simple organic substances. Further on, they are being acted upon by the decomposers to perform the process of mineralization, or we could say humification as well. That's the digestion process in which complex organic substances are now getting converted into simpler inorganic substances, right? So fragmentation is the process. Now coming to leaching, this is incorrect. What is leaching? Leaching, one important thing that you need to remember for this is the only process in the entire steps of decomposition which does not require any living organism, right? This is a physical process that occurs. In this process, what happens is that when detritivores are actually breaking down these complex substances into simpler ones, few water soluble impurities, they leach out between the soil particles and enter into the water table, right? So this is a physical process which does not require any living organisms, right? That's a important fact about leaching, right? Then talking about humification, again it's incorrect. Humification is the process that's being performed by decomposers when these particular substances that are being digested already upon by the detritivores, these are further acted upon by the decomposers. There are still certain substances which are too complex to break, right? So that is going to form a dark amorphous solid, and that particular solid is humus. It's a rich resource of nutrients and it is basically resistant to microbial action. That is why its digestion is really slow, right? So the process of formation of humus is humification, which is being done after the action of decomposers. Then we have stratification. This could also be ruled out because stratification is not at all being utilized during decomposition. It is a structural aspect of ecosystem which talks about the vertical distribution of the species in an area, right? So this is not even included in the decomposition process. That's why we could rule out this particular option. Non-usable हो जाता है. See, humus ऐसा नहीं है that it's not at all going to be digested. It is digested, but the rate of digestion, that means the conversion of the complex organic substances into the simpler one, is really slow, right? That is why we confer this fact, or we infer it that it is become non-usable. It's not. It is still a very good reservoir of nutrients, but the digestion process is of course really slow, right? So this is how It's a really fact-based question, but somehow they can tweak a question a little bit by asking that which particular process during decomposition does not require living organism, that would be leaching. They could ask you with reference to the definitions also, right? So you have to entail this process of decomposition. Also, one more thing is that that decomposition, all the steps that are being there in the decompositions are occurring simultaneously. So we are not able to form the sequence of them. Yes, leaching hone par. That's correct, Ankul. I'm really sorry. I thought you were talking about fragmentation or humidification. But yes, after leaching, it's been entering into the water table. So yes, of course, they're not going to be utilized as such. Wonderfully said. All right. So when we talk about the steps of decomposition, they all are occurring simultaneously. So we cannot able to identify that what's the correct order of them, right? So this is how the decomposition is all about, right? Let's move ahead to another topic, another question. And that question is question number 104, which says that dash serve as an important biofertilizer in paddy fields, right? So when we talk about the organic farming, it talks about that we are going to utilize certain living organisms, right? Organic, substances, organic organisms, so that we are like reduce the dependence on the chemical fertilizers, right? Which are basically adding up into the plants, and by the process of biomagnification got increased at the level of higher trophic levels, right? So that is why the concept of biofertilizers also came into picture, because when we are using chemical fertilizers at one point of time during green revolution, we have used a hell lot of it, but the consequences of that we have faced later was lot of structural deformities, physiological deformities. In order to reduce that dependence on those chemical fertilizers, the concept of biofertilizers came into picture, in which we are going to use the microorganisms in order to enrich the soil with nutrients, right? So for this we have used bacterium, we have used fungus, right, in order to serve the purpose of biofertilizers. But when we talk about the paddy fields specifically, the important biofertilizers would be cyanobacteria, correctly depicted in option 4. Like we could use Anabena, right, Oscillatoria. These kind of cyanobacteria could be used for as a biofertilizers in the paddy fields. It's not like that we cannot use fungus, right? But in paddy fields we don't use it. Fungus like Glomus genus, the members of Glomus genus, these could be used as biofertilizers as they absorb phosphorus from the soil and could give it to the plant. So they form mycorrhizal association and provide the plants with ample amount of phosphorus. So yes, they could be used, but not in the paddy fields. Then talking about mycoplasmas, they can't be used as biofertilizers, no reported cases. Also they are more pathogenic in nature compared to being a important usable microbe for the plants. Lichens, they could also be used, right? So but over here they have been specifically asked about the paddy fields, that's why we are going to consider it as a cyanobacteria. Correctly answered by Amit, Ankul, Sagnik. Wonderful, right? Fungus specifically we won't use, but if they form the mycorrhizal associations, they could be used as biofertilizers. Or we specifically need phosphorus absorption by certain plants, then we are going to employ this Glomus as the biofertilizer, right? Is it clear, Ankul? Okay, so till that time we are going to move to next question. Hope these many questions are clear to you. In case you have any doubts, please post it on the comment box so that we could discuss it. Okay, so we have another question over here which says that mark the option which is not the significance of mitosis. So when we talk about mitosis, which is an equational division, that means the ploidy that's going to be there in the mother cell is also going to be retained in the daughter cells, right? So why we use this particular mitosis? See, every process must have certain significance in nature so that it would be retained for long. If it loses its significance, then of course that particular thing is going to be eliminated in nature, right? So same goes for mitosis. It's an important process because it will help in the growth of organisms, right? If we talk about the microorganisms, single-celled unicellular organism, mitosis is basically helping in the multiplication process, that means in the reproduction. So in terms of number, they are growing because of mitosis. If we talk about the multicellular organisms, because of the addition of more cells by the process of mitosis, they are growing, right, in size, right? And why they are being helping in growth? Because what we have seen till now, that in this process there will be no change in the ploidy or in the genetic content of the mother cell and the daughter cell. That's why the similar type of cells would be added in the organism, yielding into the growth of the organism, right? Second says maintenance of cell size. So that's also true when we talk about mitosis. Then whenever the nucleocytoplasmic ratio is going to get disturbed, that's when mitosis will occur, and it will maintain back the cell size which was usually there. Then third says maintenance of chromosome number. That's also true, because as I've told you, the ploidy in the mother cell is going to remain the same in the daughter cell. So that's why maintenance of chromosome number is also going to be there. Introduction of variations is not the significance of mitosis but of meiosis, because over there in meiosis we'll be having recombination, or we could say crossing over, which will be bringing out the variations. In mitosis, that particular event is not present. That's why introduction of variations will not be a feature of mitosis. Correctly said by Sagnik. Very good. Amit Ankul also given the correct answer, as this is the only one that's not the significance. That's why it is the correct answer for the question, right? Correctly said. So this is how you will be solving this particular question. Now let's move ahead to another one. And this question says that in final stage of prophase 1 of meiosis, right? So when we talk about prophase 1, it has the first substage as leptotene, in which beginning of condensation of chromosomal material will be observed. Right? This is the beginning of it. A short-lived phase, quickly followed by zygotine, where we could see the characteristic event as synapsis, in which the homologous chromosome starts pairing together with the help of a ladder-like structure, that's synaptonemal complex. Further, it is being followed by pectine, where the recombination occurs. So during this particular phase, recombination nodules will be formed and the recombinase enzyme gets fully activated, right? And that's how it will facilitate the process of recombination. By the end of this phase, the recombination, that means the exchange of genetic material between the non-sister chromatids of homologous chromosome, is going to be completed, right? This is further being followed by diplotin, in which we are going to see the dissolution of synaptonemal complex and we are going to see the appearance of chiasmata, right? What are chiasmata? These are basically the sites of crossing over where the recombination nodule is still intact, and because rest of the synaptonemal complex has been dissolving, right? That's why those are the places which are still going to be visible, and those X-shaped structure, that means the site of crossovers that we observe now, would be considered as chiasmata. So we could see their presence in the diplotin. Now further, in case of dikinesis, this is the last substage of prophase 1, or we could say the final stage of prophase 1. Over here, the characteristic event that we see would be terminalization of chiasmata and also This phase is characterized by the other events that we see during the prophase, right? Like for example, the disintegration of nuclear membrane, the nucleolus, the disappearance of other organelles, right? Formation of mitotic apparatus, the movement of centrioles in case of the animal cell, right? All these events would be seen in dikinesis. So this is basically representing the transition to the metaphase 1, right? So by this, if we get back to the question, it says in final stage of prophase 1 of meiosis, what we observe is terminalization of chiasmata. So that's absolutely correct, right? Clearly said by Sagnik, Ankul, Amit, wonderful. So terminalization of chiasmata takes place in dikinesis, the last stage of prophase 1. Wonderfully said, right? Bivalence and chromosome number in anaphase 1 and anaphase 2. Okay. So see, as I've told you, what are bivalents? Bivalents are going to be single entity like this. Let's suppose we are having these two chromosomes. Right? If we are saying bivalent, right? So if they are going to be like performing the synapses together, this entire structure would be a bivalent in terms of chromosome number, because there are two chromosomes, right? The name is bivalent. So two chromosomes are going to be there. In terms of chromatids, it will be 1, 2, 3 and 4. 4 chromatids are there, that's why we are going to call it as tetrad. So one bivalent is going to have 4 chromatids, right? So now what happens during here is that when you are talking about anaphase 1, in that particular case, separation of this bivalent is going to occur. Bivalent chromosome, homologous chromosome is going to be, divided into 2. So let's suppose if we are talking about this, there is only one bivalent in the cell. It's hypothetical, but let's assume that there is one bivalent in the cell. So the chromosome number in this cell is 2, right? In anaphase 1, when we are going to have the separation of it, each of the cell will have 1 chromosome each, right? Now in anaphase 2, because the separation of sister chromatids occur, when the separation occurs, those two will now be considered as chromosomes. So now the progeny cells will have a single chromatid, but the number of chromosome will again remain 1. So what is the difference? Difference is that when we talk about the initial mother cell which is undergoing meiosis 1, at that particular moment of time, it has 2 chromosomes with 2 chromatids. In anaphase 1 now they have single chromosome with 2 chromatids. That's how the reduction of ploidy occurs. With 2 chromosomes in a pair, now they have single one of them. But in meiosis 2, now the chromatid separation occurs, so the number of chromosome will remain the same, but the number of chromatid will be reduced, right? That is why it is equivalent to mitosis in terms of that it is going to have the same number of chromosomes as the mother cell. It has 1, these progeny cells also have 1. But how this is different from mitosis? That in mitosis, the daughter cells are going to receive these dyad chromosomes having 2 chromatids each, but on the contrary, in meiosis 2, we are going to have the monad chromosomes with single chromatids each, right? Is it clear to you, Ankul? Any questions? So certain times you will be asking about that how many chromatids if the bivalents are going to be this much. Let's say 10 bivalents are being present, then how many chromatids are going to be there? Just simply multiply it with 4. Or they can give you the number of tetrads and then ask you the question about that how many chromosomes are going to be there or bivalents are going to be there. Then also you can divide it with 4, okay, in that particular case. So this is how you will be solving the questions. Alright. So when we talk about the first one, chiasmata formation occurs. This could be ruled out because this could be seen during diplotin. Then second one says crossing over occurs. This could be seen during pecitine. So we could rule out this. And third one is saying recombinase enzyme gets fully activated. This also occurs during pecitine. So these 3 options could be easily ruled out, right? Metaphase 2, how many chromosomes? The same number as we are going to see in anaphase 1. So because in metaphase 1 what will happen? The same amount of, like these two, no, the double of it, because till metaphase there is no separation of chromosome occurs. So whichever is going to be present during prophase, two of them, now they are not together in the synapsis mode, but they are going to be, present on the equatorial plate. The same number of chromosome which you observe in the prophase would be the same number of chromosome you would be observing in metaphase. In anaphase, after the separation, you will be seeing different ones, right? Is it clear to you? Okay. Now let's move ahead to another question, which is question number 117, and it talks about dash act as a mobile carrier for transfer of electrons between complex 3 and complex 4 in electron transport system of mitochondria. So because we are talking about mitochondria, that means we are referring towards the mitochondrial ETS, right? So when we see the mitochondrial ETS, we could clearly see it over here. Over here, if we talk about the complex 1, right, NADH dehydrogenase, and complex 2, succinate dehydrogenase, they do not have the direct link up with each other. Rather, both of them will provide their electrons to ubiquinone, right, which is, and when these electrons are being transferred, the protons are being funneled in the intermembrane space, and this ubiquinone is acting as the mobile carrier between complex 1 to complex 3 and complex 2 to complex 3. From both the spaces, it is giving it to complex 3. Now from complex 3, it has been given to a mobile electron carrier which is present on the outer side of the inner mitochondrial membrane. This is a very important line being emphasized in the NCERT also, that this is the mobile carrier which is present on the outer side of the inner mitochondrial membrane, as you could clearly see the location of cytochrome C over here, right? So it takes up the electron from complex 3 and give it to complex 4 cytochrome C oxidase, right? So getting back to this, as Ankul, Kunal have correctly answered it, that it would be cytochrome C given in option number 1. This is the correct answer. Then when we talk about cytochrome A and cytochrome A3, right, these are being, you could clearly see the components of complex number 4, cytochrome A and cytochrome A3. These are the components of complex 4. So it has 2 cytochromes and 2 copper centers. This is also a very common question that we have seen in the PYQs, that what are the components of complex 4. So there will be 2 copper centers, UA and CUB, and 2 cytochromes, cytochrome A and cytochrome A3. So getting back to this, we have these 2 could be ruled out based on the fact that these 2 are basically the cytochromes present in complex number 4. Then coming to cytochrome B, this could also be ruled out because we could clearly see that complex 3 over here is cytochrome B C1 complex. So cytochrome B is the component of complex number 3, right? So getting back to this, this could also be ruled out. So the correct answer for this is going to be option number 1. Right, I hope this is clear to everyone. All right, now let's move ahead to another question, which is question number 122, and it says that most of the cell organelle duplication occurs in. This is a question that have came multiple times, right? So when we talk about the cell organelle duplication, why this is necessary? Because this particular process is occurring during interphase when the cell is preparing itself for division. After the division, what the cell has to ensure that whatever content it has to be equally distributed in the 2 daughter cells, right? So if we'll be having a cell like this having 2 mitochondria, let's say it has single mitochondria, let's say. So it's not gonna, have the half mitochondria to one but half to other, right? So that is why it has to duplicate it first, so that in the daughter cells, the mitochondria could be equally distributed. Same goes for other cellular organelles as well. So when we have this particular saying that most of the cell organelle duplication, this occurs in G1 phase. It's been explicitly mentioned in NCERT as the line as such that most of the cell organelle duplication occurs in G1 phase, right? Correctly answered by so many students. Ashish, Shaz, wonderful. So this is the G1 phase in which most of the cell organelle duplication occurs. When we talk about the G2 phase, over here the duplication of semi-autonomous organelles occurs. That means we are referring towards the mitochondria, chloroplast, and one more which is not a semi-autonomous organelle, and that is Golgi complex, right? These duplication occurs during G2 phase, and it is also being characterized by the synthesis of a very special protein, that is tubulin protein, which helps in the formation of spindle fibers later in the M phase. Now coming to S phase, this is also a line explicitly mentioned in NCERT that in S phase what we observe is DNA replication, a characteristic event of this phase, and this particular event does not occur anywhere in the cell cycle, whether it's mitosis or meiosis, not even in interkinesis, right? So S phase is being characterized by DNA replication. Then we have M phase. This could also be ruled out because over here the division is occurring, not the duplication. So over here we are seeing the division, actual cell division. Over here the reorganization of the cell organelles are occurring, not the duplication part, right? So this is also incorrect. So G1 phase is correct. And yes, Ashish is also right. It's also been characterized by the duplication of centriole. But centriole duplication in animal cell also occurs during interkinesis as well, right? But DNA replication only occurs in the S phase, right? Yes, so you are correct, Ashish and Sagnik, that yes, this phase can also be characterized by the duplication of centriole in the animal cell, right? So this is how you will be solving the question. I hope the session is being useful to you. You have understood all the questions. So keep practicing and keep reading NCERT. Thank you very much for the session.